Hi students, today let us see some of the details on chromatin and chromosome. We'll learn from this the difference between a chromatin and a chromosome. You may have heard this term several times during your previous classes in your lower classes. Let us see some of the details about these two terms. First, let us see what is the relation between a chromosome and a chromatin. In this figure, you can clearly see how a chromosome is derived from a double-stranded DNA. Here, you can see this is the double-stranded DNA. This double-stranded DNA helix, it combines with some of the proteins. These yellow colored proteins you can see here. These proteins are, we can go through the details of these organization, these packaging details we have to learn as a separate topic. So now just see that this double-stranded DNA helix combines with some proteins. These are the yellow colored proteins that we will see in another session. And so these proteins, see how this DNA coils around this yellow colored protein. So in this way the double-stranded DNA coils around this some proteins, and through this what happens, this much length of DNA can be reduced, it becomes condensed to some extent, right? So this much length of the DNA becomes reduced as it coils around these proteins. So this is a part of condensation. So this model we have to study in detail. We call it as nucleosome model, and here this appears like beads on a string, right? Like we have our chain. In the chain we have beads in the string. Likewise these appear like a beads on a string model. So this is the first level of condensation, and later on these becomes condensed, and it again coils to form the chromatin fiber, and this chromatin fiber again condenses in several levels and forms finally the chromosome. So this is how a chromosome is formed. The double-stranded naked DNA associates with some of the proteins, and then it condenses and coils several times. Supercoiling occurs, and it is formed in this shape, and this particular shape or this particular form is called chromosome. So this is how a double-stranded DNA coils and comes to the form of a chromosome. So that is about the relation, and the more details are represented here in this figure. This is the molecular details. Double-stranded DNA, it is the base level organization. Here we can see the bases, ATGC bases. Nucleotides are being represented in this figure, and this is how a double-stranded DNA has at its molecular level. And this double-stranded DNA then combines with proteins, the same as the previous slide but represented in a different form. And these are the histones, and it coils and coils like a spring, and it coils and coils to form the chromatin, and this chromatin again condenses and supercoils to form the chromosome. So this is how a chromosome is formed from a double-stranded naked DNA. And you can see the difference. This is the chromatin and this is the chromosome. So in this slide you can see the exact difference between a chromatin and a chromosome. Chromatin is actually a thread-like structure or thread-like morphology, and here in this figure this is the chromatin, and we can have here it has a central point, that is the centromere, and when is this chromatin form seen in a cell? Chromatin is seen in a cell when it is in the interphase, that is inside the cell. DNA is not free, but it is complexed with several of the proteins to form the structure chromatin. And at the time of cell division, the chromatin becomes condensed into chromosome. So when the cell is not dividing, that is when the cell is at the interphase stage, then we can see the chromatin form. But when the cell starts division, when it prepares to divide, either mitotic or meiotic, then this chromatin condenses and forms chromosome. That condensation was the one we observed in the previous slide. So that is the difference. The interphase cell has chromatin, and the cell which is preparing for division or at the time of division, it is seen as chromosomes. And regarding the number of chromosomes, the number of chromosomes varies considerably among different plants and animals. However, each species has a constant chromosome number that we know. Every species has a constant number of chromosomes. And in the somatic cell of a plant or animal, the chromosome occurs as paired form. And each member of a pair of chromosome, we call it as homolog. We know in diploid cells the chromosomes are paired. So each member of the pair is called homolog. In case of humans, man has 46 chromosomes, or we can say 23 pairs are there, and which is the diploid number. While in the gametic cells or in the haploid condition, the chromosome number is 23. And regarding the morphology of a chromosome, each chromosome has a point of attachment, and this point of attachment we call it as centromere. It is the point where the spindle fibers come and attach. Here you can see this is the point of attachment. So this is the point of attachment or the centromere, where at the time of division spindle fibers comes and attaches to these chromosomes. So that is a centromere. The position of centromere determines the shape of chromosomes, and chromosomes are classified into 4 types according to their shape, and we can see the difference in this slide. Here this is the metacentric chromosome. In metacentric chromosome, we can see the centromere is placed at the center, and that these two arms, and these arms, both the upper arms and the lower arms are they are some most probably they are equal in size. So the centromere is at the center and the two arms are equal in size. That type of chromosomes are called metacentric chromosomes. Here it is in a V-shaped chromosomes also we can say. And in the second type, submetacentric, if the centromere divides the chromosome into two unequal arms, here the centromere is not in the exact center, but it is somewhat towards one end. So this arm is longer than this arm. The lower arms are longer than the upper arms. It is not necessary that it is always that the lower arms are always longer and the upper arms are always shorter. It can occur in vice versa also. So anyhow, the centromere is placed in such a way that any one of the arms will be longer and the other will be shorter. That type of chromosomes are called submetacentric chromosome. In the third type, that is the acrocentric chromosome, here acrocentric chromosome, the centromere is located in such a way that at one side the arms are very short and at the other side it's too long. You can see here the upper arms are too short and the lower arms are very long compared to the upper ones. So that type here, the centromere is more towards one end. So this type of arrangement or this type of chromosomes are called acrocentric chromosomes. The last one is telocentric chromosome. Telocentric chromosomes are the ones where the centromere is located at one end, exact one end. That is at the telomere. Telomere is the end region of a chromatid or the chromosome. We have the two arms, and the tip region of the chromatids are called the telomeres. So when the centromeres are arranged at the tip region, then that type of chromosomes are called telocentric chromosome. So that is the four different types of chromosome based on their shape or based on the position of centromeres.